Hello, and welcome back to Father-Offspring Interviews, episode 29. We are going to jump right into today's question. So, a viewer said, I've long wondered why discussions about intelligence and brain size reference the brain size relative to body size rather than the absolute brain size. Can you explain that?
Yes, something we have all wondered, and you do that correction to avoid an interpretive temptation, which takes you down a very incorrect path. Okay, so we're the smartest species out there, we tell ourselves in some ways. We've got the biggest brains of any species out there. Not true. Turns out there's a number of species with bigger brains. Elephants have bigger brains, dolphins, cetaceans, and stuff. Let's focus on elephants. Elephant brains have about 3 times as many neurons as we have. We've got about 80 billion. They've got about 250 billion or so, weighs 3 to 4 times as much. An elephant brain comes in around 10 to 12 pounds or so. So that's great. They've got a much bigger brain and they've got a much bigger body. But let's express that as percentages. Humans, 2% of our body weight is taken up by our brain. Elephant, a tenth of 1% of body weight is taken up by brain. If an elephant's brain was 2% of its body weight, it would weigh hundreds and hundreds of pounds. And what we see here is this wonderfully obscure term, allometry, which is the ratio of different parts, and allometry varies with size. What do I mean by this? You start with a human, and what elephants teach us is if you want to have a body about 70 times bigger than a human body, you're going to need a brain about 3 times bigger to run it. There's a ratio that is maintained there. And thus, we see across all sorts of mammalian species, there's a relationship, there's a curve. And by the time you get to elephants, triple the brain, you could run a body 70 times as big. So, what are you doing with all those extra neurons if you're an elephant? Obviously, we're going to get to you need more neurons to run a body, but you would think 3 times as many neurons. That's a huge increase. Why aren't elephants, inventing the wheel or writing essays about whether the Republican Party symbol is good or bad for the elephants? What are they doing with all those extra neurons instead? And you get an important hint that you don't have proportionately that many extra elephant neurons in the cortex. What you have instead is motor systems in the body, the cerebellum and stuff, because you have this gigantic body and you got to have a whole lot more neurons telling skeletal muscles what to do, and a whole lot more neurons telling your elephant stomach, which is the size of a Volkswagen, that it's time to crunch the food up there because there's a lot more muscle fibers that need to be innovated. What you see is a lot of the extra neurons there are simply about running a bigger body that's got a bigger everything and every organ, and 3 times as much gets you what you need out of 17 times as much. Okay, so what we see here is saying, well, what do we make of elephants and dolphins having bigger brains than us? It's an apples and oranges issue. One other domain where people think about brain size and differences in it is one where you get led into another seductive side path that, on the average, the male brain, the human male brain, is about 10 to 15% bigger than the human female brain. Aha! Say all sorts of ideologues who are ready to run with that. And then you ask a question: average body weight of males versus females, average height, average lung capacity, average skeletal muscle mass. The exact ratio differs in all of them. But when you correct for that, the 10 to 15% excess of male brain size winds up being the same when you express it per body weight. What does that tell you? That tells you that, absolute size doesn't mean a whole lot. And another way to appreciate this, okay, human male brains are 10 to 15% bigger than human female brains. Neanderthal brains were 10 to 15% larger than our human brains. And That doesn't say anything about their intelligence. Okay, so what do we learn from this? Just to tap into the message given to us by a recent American political leader, an inspirational one. All sorts of people needlessly get obsessed with the size of the brain.
All right. So, next, someone asks, is there a connection between emotional childhood trauma and autoimmune diseases? If so, do genetic factors exacerbate the correlation?
All right, one word answer. Yes, there is a relationship. Yes, genetics has something to do with this. Autoimmune diseases, there's 80, 100 different autoimmune diseases. A lot of them were well known: type 1 diabetes, juvenile diabetes, lupus, rheumatoid arthritis, multiple sclerosis, Hashimoto's disease, Graves' disease, Songren's disease. And I realize I've never actually tried to say that word before, and it's got a J somewhere in there. So it might very well not sound that way. Autoimmune diseases, what are they? It's your immune system going crazy. What does your immune system do? It attacks pathogens, external challenges to your health and well being, and gross domestic product and all of that. What the immune system does for a living is it goes out and checks every cell it encounters inside you and has a rule. Is this part of me, or is this something invasive? Bacteria, something invasive; liver, me; parasite, something invasive; eyebrows, me. It has a whole dichotomized world between self and non-self. And the immune system has to go about generating antibodies, something called acquired immunity, antibodies against invaders, against pathogenic threats, and all of that. And in order to pull that off, it's got to do two quality control things, which are so elegant. I love this. The first thing is your immune system, there's some new virus in there. Your immune system clips off a little piece of it and presents it to cells that make antibodies of all sorts of different shapes. And you then look at all the antibodies that have been made, all the antibody bearing cells, and you want to make sure you got one that recognizes this piece of this invasive virus that reacts with it. So you do this whole like assembly line of every single one of these new antibody secreting cells goes through a test. Does it attack a little model version of this piece of this pathogen? If it attacks it, that's great. If it doesn't attack it, doesn't meet the grade, destroy it, get rid of it, and you get rid of the vast majority of those cells. So that's great. So now you have a whole collection of these newly made immune cells that release antibodies that will recognize this invasive thing. But then the self versus non-self comes in, which is, that's great. You got all of these that will recognize this invader. What if this particular shaped antibody also recognizes some normal piece of you and attacks it? So what you have to do at that point is you did this first positive selective screen just to get antibodies that react against the invader. And then you got to run all of those against samples of you to make sure none of them are interacting with you at the same time. And any of the ones that interact with you get destroyed. And ideally, you then wind up with an antibody profile that will recognize only the invader. So, an autoimmune disease is when that negative selection stuff screws up and some antibodies slip through that may recognize the invader, but also recognizes a piece of you and initiates a whole attack. And it depends on which autoimmune disease. Multiple sclerosis, it's against a protein that you find in myelin and neurons. It's a neuromuscular disorder. Type 1 diabetes, it wipes out beta cells in your pancreas, so you can't secrete insulin because the negative selection screw up stumbled into releasing a class of antibodies that recognize this cell type in your pancreas in addition to this invasive thing and attack it. And the typical issue with autoimmune diseases is you have now damaged that part of your body. So we look into what are some of the triggers for generating new antibodies that put you in danger of autoimmunity. And it's things like infectious diseases and viruses and pathogens and things like that. There's increasing evidence that PASC, post-acute sequelae of COVID, long haul COVID, involves autoimmune attack on your body. Amid all of this, there's a backdrop. Lots of people got COVID. Not everybody got long haul. Lots of people are exposed to certain pathogens. Not everybody gets an autoimmune disease. Exactly as the questioner brought up, different genetic makeups make you more or less vulnerable to autoimmune diseases. Maybe what that translates into is the genetics of how likely your immune system is to screw up with that negative selection stuff. And being a brain person, I particularly love the fact there's a whole class of autoimmune diseases inside the brain having to do with the fact that normally your circulating immune cells do not get into the brain. If you learned anything about the brain for the last 3,000 years, one of the first slogans you learn is the brain is immunologically privileged. It's got a barrier that keeps circulating immune cells from out of there. Blood-brain barrier, which is remarkably elegant. And it turns out brains have their own sort of local immune system, these things called microglial cells. So you run a very high fever. And one of the things that happens with a sufficiently high fever is it loosens up this blood-brain barrier, and some circulating immune cells that are not supposed to get into your brain get in. Or you're in extremely, extremely high temperature, and there's an interesting precedent for that. And again, the blood-brain barrier opens up, and immune cells get in there. And everywhere they look, there's stuff they don't recognize because they never had to learn what, like, my nervous system looked like. The microglia do that job, and they form antibodies against something or other in the brain and attack it. And there's increasing evidence, a number of neuropsychiatric disorders, Tourette's syndrome, OCD. A subset of those people have an autoimmune disease in the brain, usually because the person had a severe fever when they were a kid, opened up the blood-brain barrier, and now you got this forever. There's increased evidence that there's a number of neurological diseases. Celine Dion fans will know that she is struggling with a neurological disease that used to be called stiff man syndrome, which was first discovered after the fevers of the 1918 flu epidemic. 2, 3 years later, it developed this disease of paralysis, which turned out the blood-brain barrier opened up, autoimmune stuff against motor systems there. But because of Celine Dion, it is now known as stiff person's syndrome. And that appears to have an autoimmune element as well. Okay, so all of that is great. So this brings us now to the issue of where does stress fit in with this? Stress, oh my God, I was so stressed this morning getting to work late. I've been stressed in recent months because of whatever. I had a traumatic childhood filled with stressful adversity. Do any of those increase the likelihood of developing an autoimmune disease? And the answer is chronic stress over the course of preceding months to years increases the likelihood. Childhood adversity, childhood stress does as well. And anecdotally, people were figuring out for decades multiple sclerosis, for example, which very preferentially attacks females, girls, and women, as do all the autoimmune diseases. And that's a whole other story about why 80, 90% of them are attacking women. But MS, it's the semester that she started college, and it was totally stressful. It was 4 years later during her first job, and she was overwhelmed. It was when that relationship, that wonderful 7-year relationship, broke up a few years after. And in the months afterward, that's where you see the autoimmune disease. So there was this long anecdotal clinical literature showing an association between major life stressors, an increased likelihood of an autoimmune disease months, a few years later, or massive childhood stressors and increased likelihood in adulthood. So, what is that about? What is causing this? And this is where one of the seemingly great long-term turns in medical history occurred. This guy, Hans Selye, godfather of stress physiology, Hungarian physician, early part of the last century. What did he live to the 1970s or 80s or so? He's the person who took the term stress from like metallurgy or engineering or something and brought it into medicine. He was totally amazing. He was one of my heroes for doing that. Apparently, he was just an insufferable egocentric jerk on top of it. But in the 1930s, 40s, 50s, he was working out the first evidence for this thing called stress, non-specific challenges, and the fact that all sorts of different non-specific, physical, psychological, too hot, too cold, starving, injured, whatever, could cause certain diseases to become more common, stress-related diseases. And he was among the first to note that lots of stress made it more likely that you would get an autoimmune disease. So he focused on one class of hormones that are central to the stress response. Everybody, when they think about stress, they think about adrenaline, epinephrine, and I look down my nose at it because it's nowhere near as interesting as the greatest hormones out there having to do with the stress response, hormones called glucocorticoids. They come out of your adrenal glands. Adrenaline comes out of your adrenals, comes out of a different part of your adrenal glands. Cortisol. Cortisol is the human version of glucocorticoids. And Selye looked at this and he said, well, an awful lot about what I'm seeing about stress causing increased risk of disease in the body is due to stress causing secretion of this cortisol, which does all sorts of bad stuff. Oh, I bet cortisol, elevated levels of cortisol will cause autoimmune disorders, will cause inflammatory disorders, things of that. And he started predicting that years and years in advance of people figuring out something very different in the 1950s, which is you get somebody with an autoimmune disease or inflammation or some, what is often done, they are put on immunosuppressant drugs, drugs that suppress the overactive autoimmunity, drugs that suppress the inflammatory response. And these are drugs, these are anti inflammatory drugs, immunosuppressive drugs, a class of them known as steroidal anti inflammatory drugs. What were these? These were synthetic versions of cortisol, prednisone, dexamethasone, triamcinolone. What people learned was glucocorticoids flattened the immune system and it became the treatment of choice for autoimmunity and all sorts of inflammatory diseases. Oh no, exactly the opposite of what Selye predicted. And Selye was nominated 17 times, literally, for a Nobel Prize and didn't get it. But the people who discovered the immunosuppressive effects of steroids got their Nobel Prize the early 1950s or so. Selye was completely wrong, totally ass backwards. Okay, but then there was another lesson in how science works. There's this great quote, Niels Bohr, one of the quantum physicist guys, who said, the opposite of a great falsehood is a great truth. But sometimes the opposite of a great truth is another great truth. And that turned out to apply here. As people learn more and more about the system, ooh, you got this much glucocorticoids in your bloodstream, and you're really, really, really stressed, and the levels go up to like here. You take 60 mgs of prednisone, and the levels of glucocorticoids in your bloodstream go through the ceiling here. They're many, many times higher than what your body ever actually generates. Jargon in the field. Instead of physiological elevation of hormone levels, pharmacological, and pharmacological levels do different things than elevated physiological levels. And what people began to learn was when you get in the range of not stress to stress, this sort, when the levels are higher, stress and glucocorticoids tend to promote autoimmunity, tend to promote inflammation. My lab did some of that work in the 90s showing Glucocorticoids, steroids, they suppress brain inflammation. It turns out it does it only in some parts of the brain, and in other parts, it makes it worse. And that has something to do with some bad things that happen with neurological disease and stuff. So, what we're seeing here is it depends. It depends on how much of the stuff you're taking. High levels, like you're stressed out of your mind, or levels like no mammal ever produces on their own, you get different things. It depends on which version. Cortisol is pretty similar to dexamethasone. It's not identical, though. They're a little bit different and they interact in different ways. It depends how long you take them for. And when stress begins to be pro-inflammatory and pro-autoimmunity, that's not one day's stress, that's chronic long-term stress. And finally, it depends where you're looking in the body. So at this point, nobody fully understands which elements of the immune system are enhanced by stress and glucocorticoids in a way that increases the chances of autoimmunity, and is it mediated directly on those hunting for invader cells or on the inflammatory. It's incredibly complicated, but what's interesting is, yeah, stress, including childhood stress, can increase the risk of autoimmune diseases. That does not mean that every person who had massive childhood adversity is going to get an autoimmune disease. It means that people who have autoimmune diseases have a higher than expected chance of some sort of stressful history starting, including in childhood. So that's the answer. And when you find out that something or other does something, at least in science, you always have to ask how much of it does that and how much is the effect that it does, because that could make a big difference.
All right. So finally, Allie from Cleveland asks, can you tell me more about how dyslexic brains work differently? Do you have any neurospicy differences?
I can't tell you a whole lot more because nobody knows a whole lot. The word dyslexia is about 150 years old, and like the amount that's not known vastly outweighs the amount that is known. Dyslexia, it's a whole umbrella term for a bunch of learning differences where you have trouble reading. You have trouble decoding letters, distinguishing them one from another. You have trouble gestalting individual letters into a whole word. You have trouble breaking the sounds of a word into its separate phonemes, or taking separate ones and being able to gestalt it into entire. It's a whole hodgepodge of ways in which brains are not very good at learning to read or learning to decode language. Roughly 3 to 7% of people get diagnosed with it. Best guess is about 20% of people have some sort of dyslexia, mild or otherwise. So we got our usual picture. 3 to 7% are the lucky ones who, when they were 7 years old, had a parent who said, we're going to go get them checked out, rather than just concluding they're not very smart or not very motivated or whatever. So the lucky ones get a diagnosis, but it is quite common. So, what is going on with dyslexia? And there's a whole history of it that has gotten more and more sophisticated. What's wrong with dyslexia? If you're looking at words and the small case b and the small case p sort of flip over on each other, so obviously there's something wrong with your eyes. First theories that came along, something's up with the eyes, something with binocular vision for accurate depth, not quite aligning correctly, or around your eyes, there's a whole circle of muscles called the oculomotor muscles. And what they do is they move your eyes around, including involuntarily in very short little jerks called saccadic motions. Not like cicadas, cicada with an S, saccadic motions, and your eyes jerk little microseconds of moving, and you turn this into a smooth perception of things. And there were immediately theories that maybe something's weird with these saccadic jerks and they're unsynchronized between the two eyes, whatever. Okay, so a little bit of evidence for that. There often are eye tracking problems in people with dyslexia, but the general sense is that's secondary to other stuff. People then moved into the brain. Your eyes see something, the information goes from your retina to your optic nerve into a part of the brain. Here, I am required not to have my license taken away. to say it goes to a part of the brain that is the sensory way station of the brain. Auditory information comes in here, and this area sends it on to the auditory cortex. Visual information comes into a different part of it, sends it to the visual cortex. This way station is called the thalamus. And there's a part of it that's the way station for visual information called the lateral geniculate. So people immediately started wondering if there was something wrong with the lateral geniculate, in particular, the lateral geniculate then passing on visual information to your visual cortex. And people found that sort of the activation of the two areas was a little bit uncoupled in people with dyslexia. So things can fall between the cracks. There were some studies that suggested that there were shortages of a certain neuron type in the lateral geniculate. Trivia, something called magnocellular neurons, suggestions of that, a little bit of evidence there. Next outpost, something with the visual cortex itself. And that probably is playing in a role in some of the most pedestrian of the dyslexias, where you have trouble telling the difference between a circle with a line going up, a B, or a circle with a line going down, a P sort of thing. And that's just the more primitive parts of your visual processing system. That's telling you, is this a straight line? Is it vertical? Is it horizontal? Is it an angle? Is there a line connected to? Are we looking at the letter L? Are we looking at the letter F, or whatever I'm doing here on that level? So some of it's that. But the senses, the most interesting stuff is coming downstream from there. Your visual cortex is telling you, like, you're looking at vertical lines and diagonal lines, and maybe it's telling you that's a letter A, and you can do simple stuff like that. It then passes the information on to the rest of your cortex, where you do analytical stuff and memory stuff. And that's where you figure out whose face this is. That's where you're able to read, when people are tracking the eyes, the first quarter of a word, and you already know what the whole word is because you could put stuff. That's the fancy stuff. And the sense in the field is that's the area where you have the most significant problems in dyslexia. Some evidence for it. There's some suggestion that some of these downstream fancy interpretive cortical areas have a shortage of neurons. People in the business, less gray matter in those parts of the cortex. Really nice work by a guy named John Gabrielli at MIT, an old colleague of mine, looking at, using very fancy techniques post-mortem, looking at the projections in these downstream areas in brains from people who had dyslexia, people without, and seeing that there's wiring problems going on there in these downstream, just the wiring's a little screwy, or the visual cortex has trouble telling the difference between, this and this kind of thing, or your thalamus is kind of sloppy and drooling in the information it sends to the visual cortex, or something, the eyes just jitter in a way that make processing difficult. Okay, so a whole array of things, whole array of possibilities. Amid that, there's a gigantic confound in the field. So you take a 20-year-old who got diagnosed with dyslexia and they've struggled with reading their whole life, and you want to see, well, what's different in their brain? So you take a 20-year-old with dyslexia and you take a 20-year-old without dyslexia and compare them. Uh-oh, no, not so fast, because among other things, the person without dyslexia, almost without exception, has vastly more experience with reading than someone with dyslexia does, because it's slower. It's a struggle. You don't do it because you enjoy it as a hobby, whatever. So maybe if you see a difference between these two 20-year-olds, it's not reflecting something about the brain that caused the dyslexia. Instead, it's a consequence of how much reading they've done over the years. So the appropriate control is whatever reading level this individual is at with their dyslexia, and a control individual at the same level of reading expertise. And thus, what you're often doing is comparing 12-year-olds with 7-year-olds and things like that. So when you actually do that more correct comparison, some of the hints of what's been going wrong there disappear. That's ruled out some of those. So that's a really important confound in the field. Neurospicy. Am I neurospicy? I have a very, very mild dyslexia. There are two letters that I reverse when I'm reading, when I'm writing, when I'm hearing, when I'm speaking as well. Not consistently, but with some frequency. And it's weird because the two letters don't sound the same. Structurally, in terms of lines, they don't look the same. They're not close to each other on a keyboard. I have no idea why my brain does this, but I tend to reverse the letters F and W. What's that probably about? The only marker I have is, in terms of handedness, I'm what's called mixed handed. I write with my left hand, I do sports with my right. 5% of the population. Mixed handedness is associated with much higher rates of dyslexic type problems.
Yeah, and just to sort of like riff on that, it manifests in really different ways. For example, I studied piano very seriously for many, many years, and still like 8 years in, I had to go through the mnemonics of, okay, every good boy does fine. It's an arduous process to sight read because my brain, I just cannot keep track of the lines, and I still do that to this day. So
Totally, you simply could not automatically decode notes on a staff. And it turns out a lot of people with dyslexia, a lot of people, or some people without any other type of dyslexia, have a music type dyslexia of exactly that type. There's been studies, if you have the lines and the staff further apart, fewer mistakes are made. If you have the stems on each of the notes pointing all in the same direction, that makes it easier. Interestingly, one study showed that if each of the 5 lines in the music staff are different colors, that doesn't help. And what you see is exactly the mistake you would expect. Here's a note sitting right on this staff line, and someone will make a mistake for the note being here or the note being there, or a note between these two staves, it's mistaken for that. So that occurs. I found a paper, 1962 paper, showing that you can get something akin to dyslexia in people who were Morse code operators. What's that telling us? Music, writing, hearing, Morse code, all of these are incredibly downstream cortical processes of pulling information out of symbolic systems and learning how to do shortcuts. So when you read a word, you don't have to read it one letter at a time, and you can hear a chord, an entire chord, and know it's a major chord or a minor without having to sound out each of the notes or whatever people do with Morse code. Dyslexia, in this broadest sense, seems to be challenges at efficiently decoding really fancy symbolic systems of meaning.
and that is it for episode 29. So keep submitting questions at the form found in the Instagram story highlight and bio or the YouTube video description. I'm Offspring Sapolsky, and thanks for your continued support of Science and the Beard.